what I'm going to try and teach tonight is a discussion of the gut. And we're going to focus pretty much on the gut only, the immune system and other parts related to it. But the dynamics of it is what I want to bring home for everybody, is that there's nothing static about our lives. Everybody moves every day. We walk, we talk, we do stuff. Our bodies are working, constantly moving. So the thought process that we have, and I've always had in medicine, that there's a pill for an ill, there's a treatment for a response, our bodies are never static. So how can you imagine that one pill with one dose for the rest of your life makes sense? It generally doesn't. And so we're going to talk a bit about this and what the gut is. Ansel Adams, one of my favorite photographs that he did. So I titled the lecture, The Gut, the Microbiome, the Environment, and Life. Disclosure, I have no financial disclosures. All the money goes to my wife if I get any. The credit, some slides are being used with permissions of Drs. Hanaway and Mullen from the Institute of Functional Medicine and Dr. Dave Rakel from the University of Wisconsin. I would also like to thank Dr. Jeffrey Bland and Dr. Weil for their genius, two great teachers who've had the privilege of learning under. The goals of the talk, we always have to have some goals. To understand the microbiome, what it is, how it functions, to understand immunity in relation to the microbiome, understand mice and the epigenome, and then understand food and the gut microbiome, because I'm convinced and have been convinced for quite some time that the healing of the human body is best done through nutrition and lifestyle management, not through pills and different versions of Band-Aid therapies. Personalized health. So I'm just going to read this because it's a little bit difficult. The mammalian gut microbiota interact extensively with the host through metabolic exchanges and co-metabolism of substrates. They conclude that appropriate consideration of individual human gut microbial activities will be a necessary part of future personalized healthcare paradigms. Why such a great emphasis here? You look at this and it's microbiota, it's the gut. We've always thought that the gut was only functional for absorption and digestion. That's what it's there for. Nothing really else matters. And we're very wrong, as usual, in medicine, that what we think is set in stone is generally not so, as we find out in the future as new studies come out. Alessio Fasano, one of the great researchers in the field of celiac disease at the University of Maryland, said this: a paradigm shift in the pathogenesis of autoimmune diseases, and for those who don't know what that means, that's type 1 diabetes, Crohn's disease, rheumatoid arthritis. In general, I even lump asthma in there, even though it's technically not autoimmune. Genetic susceptibility of the host immune system to recognize and potentially misinterpret an environmental antigen presented to the GI tract. And I would submit that it's presented to most areas, especially the lungs, too. The host must be exposed to this antigen. The antigen must be presented to the GI mucosal immune system following its paracellular passage from the intestinal lumen to the gut submucosa. What does that all mean? In other words, you must have intestinal permeability or a leaky gut, a word that 10 years ago was anathema to a GI physician. If you said the word leaky gut, they'd laugh you out of the room. Now you go to a GI conference, they're finally talking about intestinal permeability and leaky gut because it exists and we know it exists. We've known it's existed for a long time by what's happening to our patients. But just like celiac disease, until we had an antibody test to prove it, people would poo-poo the fact that people would react to gluten. Now we have an antibody, so now all of a sudden celiac is a disease. The absence of a test does not prove the absence of disease. And the absence of data that leaky gut existed did not prove the absence of that actually happening. So now we have the data. Alessio Fasano was one of the first to look at it. And these events are what precede disease. And I'm going to submit to you tonight, if you end this, after the end of this talk, you may have a further knowledge of understanding how the gut is the root cause of all of this. Everything starts and ends in the gut. Pretty much that is what I believe at this point. 5 years from now, it may change. But right now, it looks like that's where a lot of the data is. Let's look at surface area. What do we have that's exposed to the outside world? When I went through medical school, the skin was the largest organ in the body. Your skin is where most interaction with the environment happened. Well, let's look at that. Skin is down at the bottom, lung dramatically bigger, and the GI mucosa is the most exposed. And actually, if you look at it, you take somebody's intestines, which are on average 28 feet long, stretch them out, they cover the surface of a tennis court. That's how much area is exposed to the outside world. Surface per square meter, 200. You compare Lake Tahoe to Lake Norman. Lake Norman we know locally. Lake Tahoe on the map is a big lake. Surface area, or what we would call shoreline of Lake Tahoe and Lake Norman, are almost the same. Because Lake Norman has all these invaginations because it's a dammed river. The immune system. We're going to talk a bit about the immune system. I'm going to try not to bore you with too much science, but we're going to talk about that. 70% of the gut is where the immune system is located. 20% in the respiratory tract, 5% in the vaginal locale, and then 5% in other places. Why is there so much in the gut? Well, the answer is pretty simple. Look at how much surface area is exposed to the outside world. The immune system wants to recognize and understand the outside world and live with it. not fight against it by nature. It's not taught to fight against it. It's taught to fight with it and live with it, and then only respond to pathogens that are dangerous. Prokaryotes, okay? We look in our bodies, and now we've realized this through a lot of the DNA analysis, that in our bodies there are 100 trillion prokaryotes. There are 10 trillion eukaryotic cells, okay? That's 10 to the 1 more foreign DNA than our own. What are prokaryotes and eukaryotes? Well, prokaryotes are bacteria, viruses. These are cells that don't have major organelles like nuclei. Our cells all have nucleus and very formed organelle systems that help us function on a more difficult level, per se. There's a lot of DNA in our body that's not our own. Let's talk about the neonatal gut. When you're pregnant and you have a child and you carry that child to term, when the child is born, the child's gut is sterile. There are no bacteria in there. The bacteria only enter the child's body after delivery. Well, what happens? Well, you're bombarded immediately. You come either through the vaginal canal or a cesarean delivery, and bang, you start to get exposed to microbes. You're either fed with a bottle formula with a nipple on it, or you go on the breast and you get bacteria that way. You get exposed to bacteria by a doctor or a nurse who handles you with gloves. You get exposed to chemicals that may be floating in the air. All of a sudden, things are starting to enter the system. Gut microflora inoculation depends on all of this stuff, and we're going to talk a bit about that. Squatters' rights. You know, when somebody gets on the land and they're there first, they generally believe it's theirs, and they hold on to it. Well, same thing happens in our gut. Microbes, the first ones there, tend to proliferate and tend to own the space. So if you're delivered in the OR, you're more likely to have Staph aureus than you are if you come through the vaginal canal and get lactobacillus. We never thought that was much of a deal. It's a big problem. It's very difficult to change these guys. When they take hold, they do not want to leave. Okay? We're learning now, and there's data now showing, and I'll go into this a little later, but fecal transplants. I don't know if anyone's ever heard of this. Fecal transplants are taking stool from a human that's healthy, taking it, microfiltering it, and then giving it to somebody via enema, and hopefully changing stuff. And there's data now showing that that helps. That'll change the flora. Changing your diet long-term can do that too. But squatters win. So what we want to do, we want to think about how do we get this under control from the beginning. Well, let's look at what happens at birth. There's nothing in there. So preterm, full term, mode of delivery, maternal weight, maternal diet affects it, hospital environment, contact with the mother healthcare staff, antibiotic use, feeding choice. All of these things affect what microbes get in there. The diversity happens over time, and you watch up until 24 months. Things become a little more interesting. Now you have, are you still breastfeeding? Are you getting exposed to good bacteria externally? Probiotics, we call them. Prebiotics, these are foods that the good bacteria like to feed on, or bad bacteria for that example. Antibiotic exposure, we're going to talk a bit about that. That's a big problem. It has been for quite a while. Timing of weaning, foods chosen, home structure, number of births, hygiene hypothesis. All of these things affect what bugs end up in your gut microbiome. Little cartoon I like, human breast versus cow breast. You know, you show over here which public parenting behaviors come under attack. You know, for years, breastfeeding, you're not allowed to do it in public. It's frowned upon, all these bad things. But, you know, people spanking their kids, not such a big deal. You know, we're big as pediatricians into moms breastfeeding, and I'll talk a little bit about why. We want to inoculate the gut. We want the gut to have the right bugs in there. We want to find out the best way to get there. We look at data. Formula-fed infants support a higher volume of Clostridial species and Bacteroides species. Other data shows that increasing in those species increase your risk of atopic disease. So until we knew why it was happening, they actually figured out that there was an association between the gut having Clostridia and asthma or eczema. Okay? Neonatal and postnatal antibiotic exposure alters this gut flora. They've done studies to look at that. If you give antibiotics to a kid and then check their gut flora afterwards, you change them artificially. 6 months to get E. coli to colonize the gut, and 80% of kids today used to take weeks. Now they've done studies showing because of different various reasons of exposure, kids don't get normal bugs in their gut till 6 months of life. What happens immunologically? What happens genetically? We're going to talk about that. 20% increased risk of asthma if you're born by C-section. C-section rate was getting up into the 30% range in the United States at one point, higher in other countries. So let's look at the frontiers of cell infectious microbiology from 2012. They looked at DNA analyses and said, okay, what bugs are in your gut? At birth, none. At baby, you see it shifts based on breastfeeding versus formula feeding, different microbes. If you get solid food, all of a sudden the microbes shift again. As you get older, toddler, if you get on antibiotic treatment, look at the vast difference between antibiotic treatment, malnutrition, and the kid who's not. Now, we're not submitting to you what's happening yet. We're just saying there's differences. Over here, you have healthy and obese. Get older, things change. A lot more purple, a lot more firmicutes. And I'm going to show you later how firmicutes are actually problem players as they get older. So what affects the gut microbiome? I'm going to list the major players. Genes, age, probiotics, drugs and antibiotics, infant environment, maternal flora, prebiotics, the food that feeds the good bugs, mode of birth, and most importantly, diet. Okay, there's many others, but these are the big players in the game. Let's look at the alimentary canal. Start out in the stomach, acid, pH. Very few bugs can survive there. Maybe H. pylori, a few others. Everybody else dies. Too acidic. As you come down, you start to see bugs show up. You see the green bugs. You see the orange bugs, the blue and the gray. And the orange there is nutrients. And they're coming down, and you see it starts out sparsely, very few bugs in the small intestine. As you come around, they start to increase in the small intestine, get past the ileocecal valve, and now bang, got a lot of microbes. 100 trillion. Think about that number. It's a monster amount of number. But again, when you have a surface area of the size of a tennis court, those bugs have plenty of place to hide. And this all affects us. Let's talk about current therapy. What's happening in the world right now? How about a mother who eats purely pro-inflammatory American food during pregnancy, takes Prevacid to help her diet-induce gastroesophageal reflux disease. She delivers by C-section because she scheduled it for some reason, receives antibiotics for group E strep, is stressed out, and then uses cow-based formula for the first year. Then the child goes out on a standard American diet, visiting your fast food place of choice and getting sweet drinks, and is exposed to multiple rounds of antibiotics because he's getting sick. Ear infections, various things, pneumonia. Epidemiology, the data is showing that each one of these things has independent risk factors. And we're doing all of them. And you wonder why disease is on the rise everywhere, and you wonder why obesity is on the rise, and you wonder why so many things are wrong. We're doing this to ourselves. Let's look at this from a paradigm. Infant species poor and stable. They're born, there's nothing there, and it slowly increases. And then over time, positive feedback occurs until you get to a healthy, stable state. Good bugs in there that do the right thing and keep us in good shape. Up at the top, you see resist colonization by bad bugs. So you're in good shape. All of a sudden, something happens, a stressor. Acute disturbance, we call it. Antibiotics, ear infection. You get a degraded state. The bugs change. All of a sudden, we're in trouble. So maybe there's a complete recovery. Maybe you got probiotics while you're on this. Maybe something else happened and you went back to normal. You're still good. Well, let's say by chance you didn't. You had an incomplete recovery. Now you're in a place where the bugs are different. They're not as stable. Things aren't as happy. Now you start to see other issues come in, but then those guys are taking hold. What are they doing? They're preventing the good guys from getting in. So now you have this unstable environment. What does it do? Well, let's go back in time. Hygiene hypothesis. When I first started in Salisbury, I was really interested in hygiene hypothesis, which was the work done early on that looks at people who are exposed to microbes early on have less disease. And this has been done in multiple different studies looking at this, but let's just take farm living versus city living. They looked at France and compared people who lived in the cities, same ethnocentric population, and people who lived on the farms, and just compared disease, specifically atopic disease. They looked at asthma, allergies. Huge decrease in the folks that are on the farms. So these guys at this time didn't know any better, but they were positing that this must be related to microbial exposure. People who lived on farms had more cows, animals who had poop lying in the field, get it on their boots, carried into the house, kid gets it somehow, and next thing you know, their microbes change. East and West Germany, everyone thought, oh, when they do this data, East Germany is much dirtier, it's going to have worse disease, more atopic. It was the opposite, flip-flop. East Germany was dirtier before the reunification with West Germany, but they had less disease because they had more microbial exposure. Germany being such a sterile environment in the west side, they didn't have the same microbial exposure, so they had more disease. And you're seeing this huge epidemics of problems in the northern Scandinavian countries, which are very clean by nature. Issues with type 1 diabetes and celiac disease. We talked about the delayed colonization of E. coli, but this is specifically happening because of C-sections. We're seeing that. This changes immunologic stimulus patterns. We're going to go into a little bit of science right now. We're going to talk about immunology. I'm not going to bore you, but I want you to understand how the differences occur. And there's just a little slide that says, I'm fed up with this guy, let's become pathogenic. Bugs change. They're dynamic. They mutate. Well, what is our immune system composed of? When I was in medical school, it's composed of two types, T helper cells 1 and 2. And they're built and set up into place to respond to environmental signals that are problematic, specifically bacteria, viruses, parasites. And so we look at this, and very simply, when you're born, they're TH0. They don't know which way to go until they're stimulated. So something comes along and says, okay, you're a virus, let's go TH1. You're a parasite, let's go TH2. You're a bacterial defense, let's go TH17. And so they all differentiate based on their exposures. The balance is the key. Everything I teach in life with these kids I see in the office is balance. If you stay in balance, nothing goes wrong. If you tip one way or the other and too much of anything, something will go wrong. And the immune system is being tipped in the wrong way, one or the other, and disease is coming because of it. Mom is Th2 while she's pregnant, because if she was Th1, she would kill the baby, because half the genes are dad's. Turner syndrome aborts 99% of the time. What happens is when the genes are figured out to be abnormal and the child's not going to be normal, the body shifts to a Th1 paradigm, kills the baby, aborts, and it works. So the mother stays Th2 if it's a viable fetus. It's all good. So when mom's born, baby comes out Th2 type with his Th0 basis. And then from that day on, that child's genetics and exposures in the environment dictate what's going to happen. Balance would be key. We thought cleanliness was better. We're wrong. It's really complicated. The immune system is something that I wouldn't even begin to try and explain to you all. I can barely wrap my head around most of it, but just let you know that it is all talking to each other. And each part of the immune system knows what different parts are doing, and based on their exposures, dictate what's going to happen. Let's look at the gut. This is a beautiful slide from Abraham et al. At New England Journal of Medicine on the inflammatory bowel disease. This is basically a cross-section of the gut at a microvillus layer. So this is a single cell layer that just goes up and down, all the way along the bowel wall. That single cell layer of gut is layered by mucus, has bacteria and food in the luminal structure, and then right below it, here's your immune system. 70% of your immune system sits right there. Why would 70% of your immune system sit right there? Defense, right? If you have so much exposure to the external environment where bad guys are, we thought the world was full of pathogens, bad guys are there, we have to have a defensive mechanism. We have to have the Army, Navy, Air Force, Marines ready to roll. So we have defense. What are the mechanisms? Mucus layer, physiological barrier, it's just a wall. You have secretory IgA. These are immune system cells that are sitting there ready to respond to whoever's sitting there. Microflora, those are the good bugs. They could be bad bugs, but they're all sitting there. Malt and gall, mucosal-associated lymphoid tissue, gut-associated lymphoid tissue. These are all the cells ready to respond and fight. And I like the slide here. It basically says, hey kid, want to be a superbug? Stick some of this, stick some of this into your genome. Even penicillin won't be able to harm you. It was on a shortcut through the hospital kitchens that Albert was first approached by a member of the antibiotic resistance. There's truth to that. They're doing this. They are really changing genes to be able to handle whatever they want to decide in life to do, to fight off our antibiotics and different things that we've done to them. Malt and gall. We're going to talk a little bit about this in brief. Mucosal-associated lymphoid tissue. So here you have your little beautiful structure of the GI tract, and right underneath it are all these cells sitting here ready to respond to the environment and develop tolerance. And we're going to talk a little bit about tolerance. The gut-associated lymphoid tissue is the other side. A little bit more here. These guys have the ability to do similar things as well. Immunologic actions of the gut flora. So in that mucus layer, there's all those bacteria that we have that are good and bad. What do they do? Well, look here. They inhibit pathogenic bacteria. So the good guys in there are doing us a favor. They decrease the pH. They decrease epithelial binding. They increase defensins. They provide food. They make something called short-chain fatty acids that actually feed our cells. Without bacteria, our enterocytes in our gut can't survive. They need bacteria. It's an amazing revelation. They increase mucus, increase healing, decrease cell death, increase barrier integrity, and then in immunoregulation, there's data showing tremendously that good bugs down-regulate our immune system. Tell the immune system to quiet down, relax, don't be so out of control. Hygiene hypothesis is basically on the premise that there's not enough bacteria that are normal to work on and learn from, so the immune system's sitting there sort of mad. It's like having an Air Force with no military police. What do the guys do? They eventually get in trouble. They're sitting there with their guns and stuff, and they're frustrated. They have nothing to fight against, so they get in trouble. So same thing happens in our gut. If we don't control it, these guys get out of control. The new data is showing that our guts are permeable. They shouldn't be. And I'm going to show you the cell structure, why it shouldn't be permeable. But what's happening now is our gut is becoming permeable to bacteria and food proteins. Everybody heard that peanut allergies are on the rise, right? Everywhere you turn, peanut-free school, peanut this, that, and the other thing. Well, that doesn't happen unless that peanut got from a place where it was, in your mouth, in your gut, to a place where it shouldn't be. It ended up in the immune layer, underneath that mucosal layer, and all of a sudden the body said, oh, you shouldn't be here, you must be a virus. Let's kill you. So every time you eat peanut, what happens? Your body tries to kill the peanut. Instead, it kills you. So we had something go wrong, gut permeability. What is gut permeability? It's the increased intestinal permeability is an early biological change that often precedes the onset of autoimmune disease. This is from Menard's work. and mucosal immunity. We know that genetics are involved. First-degree relatives of patients with Crohn's disease. That's an autoimmune disorder of the gut. First-degree relatives of people who have Crohn's disease have leaky guts as well. But they don't have disease yet, but they have leaky guts, so there's a genetic component to this as well. Something's going wrong that's causing this to happen. Let's look back at this structure again. So again, you have this beautiful mucus layer, and then you have now control here where these immune cells are sitting here ready to taste the environment, learn what's right from wrong, send down signals here to the lymphocytes and say, okay, become Th1, Th3, Th17, fight off virus, bacteria, all these things. Do your job. That's what it should do. What happens when it goes wrong? So you see the normal side over here. Now you see over there the mucus layer is disrupted. All of a sudden you see the cells are starting to die. So this hole's now developing. Well, all of a sudden, those red things that shouldn't be in that space below come below. The immune system sees it and says, time to go after you. So all of a sudden, you see the change. Now you have clonal expansion of all of these cells. They go after it, produce all these antibodies. Those antibodies now are ready to attack that perceived pathogen. It's not a pathogen. It's wheat. It's peanut, right? We hear about all these allergies. These are problems. Going back to hygiene hypothesis, we're going to look here a little bit and see. There's this thing called a dendritic cell. That's this little cell right here. This is just beautifully fascinating. This cell actually snakes its way up through. That is 9 microns wide. I don't know if anyone knows how small that is, but that's unbelievably small. Electron micrograph's the only way to see it. This little dendritic cell will send his little foot up there to taste the bacteria as they come by, because it knows nothing's supposed to leak through it. Tastes the bacteria, learns it, says, you're a good player. You don't have to worry about you. So this was set up from the beginning to learn right from wrong, what we call tolerance. Tolerance to the environment, tolerance to good things. Those tight junctions there is what we talk about. This is what's getting disrupted, tight junctions. And then when the cells die because of all the inflammation that occurs afterwards, now you have not even the tight junctions are leaking, but everything's leaking. And then the commensal bugs. We're going to talk a bit about the probiotics again. The good guys that should have been in there first, the squatters' rights, the lactobacillus from the vaginal delivery. You have mnemonic lactobacillus labia, or the bifidobacter that comes from breastfeeding, bifidobacter breast. These are ways, those are the bugs that should be there from the squatter's age. But instead of, because we C-sectioned and formula fed and antibiotic treated these children, they don't have the good guys. So they have no protective barrier. What causes some of this stuff? Abnormal gut flora causes poor tight junction function. Poor nutrition, decreased short-chain fatty acid production, so our enterocytes don't have food, so they don't grow right. And acid use increases pathogen survival. If you raise the pH of the stomach, all of a sudden more pathogens can make it through to the small intestine. Also, proteins can remain intact. So the peanut protein, instead of being hydrolyzed a little bit, stays intact, so it has a chance to leak through. NSAID use. People use NSAIDs all the time. Ibuprofen and naproxen. These are drugs that are commonly used for pain. They uncouple that tight junction. They actually physically open the muscle, let it come wide. You don't hear people ever talking about that in the commercials. It's a problem. Ethanol abuse, so, alcohol. Antibiotics, of course, increase pathogens because you select out killing off a bunch of guys. Somebody else is going to grow back, and if it's a bad guy, it's a bad thing. So what? Doesn't matter. We've talked forever about leaky gut, but nobody says it's a big deal. Should we care? Are we all the same? Are we all different? Do we have the same bugs? Are our bugs different? We're all individuals. Everybody looks different. We all act different. So why shouldn't we have all different bugs? Well, let's look at some of this. Reinhardt, in 2009, looked at what's the colonization pattern of developing human gut. Well, early on, you have proteobacteria, which is E. coli, and then those guys start to go away as the oxygen level in the gut decreases. And then you have these firmicutes, which are streptococcus species. Then you have Bacteroides and Actinobacter. So those are the guys that come in. Names that don't mean anything to you guys, but they will as the data starts pouring out and showing us. Enterotypes, these are the gut types, the bugs that are in there. Dietary habits are linked to the type of bugs that thrive in the gut. People who eat a lot more meat and saturated fat, Americans, tend to have more Bacteroides in their flora. Ruminococcus prevailed in people who consumed a lot of alcohol and polyunsaturated fatty acids, whereas Prevotella favored a diet rich in carbohydrates. So here you have a study that's basically showing what you eat is what you get. You eat the wrong foods, you get the wrong bugs. You get the wrong bugs, you have disease. So we've always said, food doesn't matter. I went to medical school. Nutrition was a class that you blew off. Nobody really spent much time studying it. When I left medical school, I knew nothing about nutrition. Zero. My wife's a nutritionist, and she laughed at me when we would talk about nutrition. She'd be like, don't you guys know anything? And no. That's why I consult you. And that's sad, and it shouldn't be that way. So when I went back and did my fellowship, I started learning nutrition, and lo and behold, that's going to be the future of medicine. If people will listen, therein lies the hardest part. So let's look at the benefits of genetic richness. So if you're not obese, you are likely to have a humongous microbial complexity in your gut. You will have tons of different types of bugs with different DNA analysis and everything. If you are obese, you have very few. Metabolic disease risk goes up if you have a few bugs. Metabolic disease risk goes down if you have a lot of bugs. Dietary intervention goes up if you have few bugs. Why? Because if you give a dietary change, you can make those bugs shift to more complexity. It doesn't happen overnight, but it definitely happens. And so here's proof to that effect. Linking long-term dietary patterns with gut microbiome enterotypes. And there in the beginning it says, diet strongly affects human health, partly by modulating gut microbiome composition. And I love the way they put by partly. There's nothing that does everything. We are a symbiotic creature. Everything we do affects everything else. So diet is just one piece of the puzzle, but it is a monster piece. Fat bugs. This was from Nature. They looked at this and said, obesity in the gut flora. That's where all the data is going now. Chemicals are a big part of it too, but there's a huge amount of influence in looking at what's happening in the gut. Firmicutes. Remember we talked about that in the beginning there, where that went way up, and you notice firmicutes get more and more as you get older? Well, firmicutes are associated with obesity. Bacteroidetes are associated with being non-obese. Impact on metabolism results from the imbalance of these phyla, these bugs, or is it an effect? Is it a cause or the effect? We don't know perfectly yet, but we know that when they're there, they're a problem. More data is going to keep emerging as to which is first. Worlds within worlds, evolution of the vertebrate gut microbiota and obesity-associated gut microbiome with increased capacity for energy harvest. Article after article after article. I decided in this lecture I wasn't going to just list article and read the study and tell you about it and read the study and tell you about it. It's not interesting. There's tons of them. Google gut microbiome, and you can be a year's worth of reading on the first night. You can just sit there and read and read and read, and there's so much data pouring out. Let's look at mice. Bacteria, diet, and obesity. So germ-free mice. Mice that have no guts, no gut flora. They're just sitting there ready to go. So let's inoculate those guts with bugs from an obese mouse. What's going to happen? Well, they gained a lot of weight. Fed the same food. You give them bacteria that are different in their gut, and they gained a ton of weight. So what does that tell us? The bugs change how we metabolize food, how we store food. That's a scary thought. Germ-free mice inoculated with microbiota from lean mice, while on the same diet as the other guys didn't gain the same amount of weight. I mean, process that. All of a sudden, the gut flora that we have dictate whether we gain or not gain weight. I mean, who would have thought that 10 years ago? I mean, it's almost scientific, like doo-doo-doo-doo. I mean, it's weird. Burkina Faso children showed a significant enrichment in bacteroidetes and depletion of firmicutes. Firmicutes associated with obesity and metabolic syndrome. Kids in Burkina Faso eat what? Vegetables, fruits, local food. They don't go and get a ring-ding-ding-dong, whatever it is that we eat around here. European Union children had a different species, and it was mostly stuff that's associated with more problems. Irritable bowel syndrome. Everybody's heard of irritable bowel syndrome. 20% or more of people at some time in life have irritable bowel syndrome. Right? That's a dysfunction of the gut where you have abnormal defecation with either diarrhea or constipation, lots of cramping, lots of complaints, but no actual physiologic problem to the extent of like Crohn's disease where you have all this inflammation. But you feel bad, right? Well, they looked at this and said in children with irritable bowel syndrome, what bugs do they have? Well, it turns out they have a lot of ruminococcus. They shouldn't, but they do. Okay, so there's something that's potentially treatable. Instead of looking at kids with irritable bowel syndrome and going, oh, you just need to take Levsin for your smooth muscle relaxation and maybe some hypnotherapy, we can actually maybe affect the gut. Who knows what this is? Crohn's disease. Okay, so that's a snapshot of the intestine of somebody with Crohn's disease. How would you like to have that as your gut? When I was in medical school, unfortunately, this was the one disease after reading about it that I did not want to have. I would have taken cancer over this. And I don't know if anyone has Crohn's in this room, and I hope you don't, but it's a very, very difficult disease to have in your life. And if what I'm telling you tonight is true, Crohn's disease is preventable by altering your gut microflora early on and not letting you get to this point. We found that children who had IBD or Crohn's disease or ulcerative colitis were 3 times more likely to have had antibiotic use in the first year of life. How many kids in this room, how many parents in this room have had their kids get antibiotics in the first year of life? A lot would have. Right? Kids come out in the hospital and I walk down the floor and I tell the mom, you really need to breastfeed. Are there any OBs in the room? Okay, good, because they get offended when I say this, but it is important that we tell mothers that it is a big deal to breastfeed. And I wrote the article in the Salisbury Post about this, and then a pediatrician in Concord wrote back stating that we shouldn't demonize formula. We should demonize formula. We should do it in a very polite way where if you need it, you take it, but you shouldn't want it. Similac, similar to lactation. The formula companies want us to believe that it's as good as breast milk. No way. Now we have the data to show it. Now we have the data to say it is not the same. It should never be the same. It shouldn't be allowed. The government should not be subsidizing formula, knowing that downstream we're going to have to pay more medical dollars for disease. Breast milk is free. Anyone heard of C. difficile colitis? You take antibiotics. Unfortunately, then sometimes C. difficile, which is a bad player, grows up. We've gotten to the point now where some C. difficile colitis is untreatable with antibiotics. People die from this. Well, in Boston, they're doing studies now where they take people with C. difficile colitis, take good stool from a healthy person and give them an enema, and within 24 hours, the previously untreatable disease is gone. Overnight. No antibiotics, no drugs, no nothing. All you did was say microbiome change. That is the most powerful thing I can remember in my 20 years of medicine, is to give somebody an enema of poop, and you cured a disease. They're doing this in Crohn's now. FDA has not licensed this for primetime live use, but it's coming. The reason the FDA hasn't made it prime time is because they've done some studies, and some people actually then ended up with secondary diseases like autoimmune arthritis, because you're giving microbes and they're shifting the genome. You can turn on disease as well as you can turn it off. So we have more work to do, but at least now we have a window into future. I love this slide. This is just an example of how it all starts. Some of the stuff we already showed, baby to adult, Bacteroidetes first, Firmicutes later. Everything starts to happen in symbiosis. Things are good. You're getting good signaling. Nothing's going wrong. Oops. Now you come down here and you start to get exposed to drugs, antibiotics, abnormal microflora start to happen. Dysbiosis, the fancy word for your gut doesn't have the right bugs. Now you start to swing into this side of the paradigm. Now you have disease. Anyone heard of H. pylori? H. pylori is the bacteria that was found that gave the 2 gentlemen the Nobel Prize in Australia for associating H. pylori bacteria in your stomach with peptic ulcer disease, right? It's the greatest thing ever. We figured out, oh man, we killed these bugs, peptic ulcer disease goes away, it's great. Do you think that was the final answer to that problem? No. H. pylori in the stomach increases acid content. It tells the stomach to increase the acid. It says, I want you to stay low pH. I want you to make your environment inhospitable to everybody else but me. It also says ghrelin, which is a signal for hunger. It says, after a meal, ghrelin, shut off. Don't be hungry. They've looked at people who got treated with triple antibiotics for peptic ulcer disease with H. pylori, and their ghrelin signal doesn't shut off. So guess what that tells them? So people are more obese after they get treated for peptic ulcer disease. Wow. Medical intervention, didn't think it through, secondary problem. Now, there's a problem. If you have peptic ulcer disease, what do? Well, something in your DNA was off, and it turns out that there's a defect in an enzyme from a genetics standpoint that gets clicked in certain people that gives them peptic ulcer disease. So we don't know what the answer is there, because would you rather be obese or would you rather live with peptic ulcer disease? It's a tough call. But there's no free lunch ever. You mess with the genome of the DNA of these bugs, you're messing with a bull's horns. Finally, common sense. This was one of the greatest things I saw about 5 years ago. Recently published evidence-based studies have confirmed that and quantitates the risk of not breastfeeding. Thus, commercial infant formula feeding should not be considered as a lifestyle choice, but rather as a basic health issue. This is from the American Academy of Pediatrics. How much press does this get? This should be on every bloody radio, every newspaper, so that it is shameful in our country to not do this, unless you don't have a choice. And I am all for it. If you don't have a choice to breastfeed, formula is your answer. But if you have the option, I had a mom sitting in my office today who quit breastfeeding at 6 weeks, and I said, why? She goes, because I got tired of waking up every hour. That's parenting today. And even after explaining that this is a big deal, if you do this, your child will be sick more often and potentially sick older. Doesn't matter. Mechanisms of health benefits. Formula-fed infants are more likely to develop a wide range of infections as compared to the rates in breastfed infants. And I had 9 slides on this from a different talk I did. I only pulled out one. The amount of data on breastfeeding and necrotizing enterocolitis and decreased asthma risk it's insane. Caesarian section. Colonization of newborn intestine with lactobacillus and bifidobacter, the 2 good players, is delayed by 30 days in those delivered by C-section. Why? Because your first bug you see is a Staph aureus. Who's heard of methicillin-resistant Staph aureus? Now you're getting exposed to bugs you shouldn't be. Your first kiss is from the OR. Not a good one. Your first kiss should be from the vaginal canal. What's wrong with our incubators? What's wrong with what's going on in the womb? Well, we're being exposed to more pesticides. Ken Cook does great lectures from the Environmental Working Group. He's been here twice that I know of. His organization does phenomenal work looking at chemical exposure, what it does. We need to avoid chemicals. I don't know if anyone read the article I wrote in the paper a few weeks back, but Liz Mumper up in Virginia did some work looking at autism. Now, whether her work is going to be proof conclusive, I don't know. But she had 7 principles of things she told her mothers to do to avoid the risk of developing autism. And there were simple things. Breastfeed. Take probiotics. Avoid antibiotics at all costs while you're pregnant and when your child's born. And she had other reasons too. Avoid chemicals at home. Try not to get exposed to things through drinking glasses. Use glass instead of plastic. Do things that make sense. And none of them are onerous. If you go on the line right now, there are docs writing back against her like she's the demon. How can you put out this data that says that this is... I don't care if she's right or wrong at this point. Nothing makes sense that's wrong in that. Every single thing in there makes sense to me to do. And until we know that she's wrong, we should be saying everybody should do it until we prove she's wrong. But that's the opposite. The medical community is demonizing people who say this kind of stuff because we don't have enough evidence. How much evidence do you need that we're going the wrong direction? Food's wrong. This is Dr. Weil's slide on anti-inflammatory diet. This is the diet that makes sense for most people. It's a bell curve. Some people this is not the perfect diet for, but it's a good one. It lists the kind of foods you want to eat. Instead of the modern American diet, which is loaded with mostly this stuff, that's in the first thing of the old pyramid on the plate. This is what we should be mostly doing. Vegetables and fruits. A little bit on top. This is available on the web. So let's think about this. We have a disease state. We've messed it all up. How do we get back to a healthy state? I just got back from San Diego to a phenomenal conference on the gut. Some of the best teachers in the country. Jerry Mullen from Johns Hopkins. Unbelievable teaching. And this was a slide I got from these guys at the IFM. And basically they're looking up here and they say, antibiotic regulation, prebiotic regulation, probiotic regulation. Let's alter the flora now. Let's see if we can shift the metabolome, the gut microbiome back to a healthy state so we're good. That's what the future of medicine is. And if you find a doctor who knows how to do this, you will heal a heck of a lot faster than you will with medicine. Eli Metchnikoff, Nobel Prize winner, 1908. Eli Metchnikoff was a famous physician who researched the function of macrophages, which are white blood cells that are able to engulf bacteria, process them up, spit them out, and put on their cell surface part of their protein, so then the body then knows if this bug ever shows up again, you're roughed up. That was his work. But he also was the guy who said, I drink sour milk every day of my life because I know the bugs in there are keeping me healthy. He knew this in 1908. Prebiotics, probiotics, we talk about this in yogurt, kefir, sauerkraut, pickles, Bubby's pickles, which is a really good one. I was talking to Chris Nagy tonight. He said to comment on Bubby's pickles. When you smell it, it's live. It's not like Vlasic, chemical-induced junk. I'm sorry, Vlasic. But it is. So let's talk about probiotics. These are the good guys. So we've gone through the whole gut microbiome and everything, and we know how we mess it up. How do we fix it now? Well, probiotics, what are they? Live microorganisms, which, when administered in adequate amounts, confers a health benefit to the human host. So basically saying, when we give you a bunch of these guys, they help. Well, you have 100 trillion bacteria in your gut. What is the normal dose of probiotic? Does anyone know? No, that's a really heavy dose. But it is. We use that. But normally it's 5 to 10 billion. So think about the number, 100 trillion, 5 to 10 billion. How the heck is 5 to 10 billion going to do anything to that 100 trillion? It does. And amazingly, it does. What are the functions of probiotics? This is where the key is. Remodeling of microbial communities. They get back in and fix the players. Take the squatters and kick them out and put some new guys in. Suppress pathogens. Improve intestinal barrier function. Get that mucus layer fixed. Affects the epithelial cell differentiation, suppression of inflammation, enhanced immunity. Enhanced vitamin processing. Without bacteria, you can't even process vitamins. How about that? Regulate digestion, facilitate absorption, enhance T regulator cell function. Remember I was talking about those military police for the immune system? That's the T reg cells. Guess what else happens to help your T reg cells function? Anyone heard of vitamin D? That's what vitamin D does. It sends a signal to the nuclear protein of T regular cells to turn on. It induces something called FOXP3. You don't get enough vitamin D, guess what? Your T reg cells don't turn on. Oops. But what if probiotics can come in and say, oh, I can help you get on. So there's multiple ways to turn them on. Go back to this slide. We have all that dysfunction up top. How do we fix it? Let's put the probiotics back in. Let's help the system do its job. This is some studies we're doing at our practice. We do some stuff that's a little off the grid. This is not ready for prime time for most clinics yet, unfortunately, because it's considered investigative. What we're doing is we're culturing stool on kids. We're sending off their poop. I do this in autism. I do this in Crohn's disease. I do this in autoimmune disease. And we look at this stuff. We say, what's in there? Well, we look at the beneficial bacteria. This kid has no lactobacillus. Why would he have no lactobacillus? Lactobacillus labia. He came out C-section. Plenty of bifidobacter, so he was breastfed probably. E. coli looks good. Oh boy, because he had no lactobacillus, not enough room, Klebsiella grew in. Nobody knows this. But we did this test and found this kid had some bad bugs in there, so we treated it. Knock it back. Yeast is overgrown. Too much of a bad thing. Everybody knows when you give a kid antibiotics or give a woman antibiotics, what happens? You get a vaginal yeast infection. Why? Because you kill off the good guys. The yeast says, woo, Cinco de Mayo. And you got a party. And so the yeast grow up. Well, there's a big debate in the community about does yeast affect us? I'm not a rocket scientist, but if something goes from 1+ to 4+, do we not think it's going to have some kind of effect? It will. We know that thrushing babies, if you don't treat it, it'll be a big problem. Why would we not assume when you get 4+ in your gut that it's not having a metabolic effect? Look at all the diseases that are associated with this. Gut-brain hypothesis, autism. There's data looking at the microbes of kids with autism, and they are way off. The diversity is low. The quantities are low. Everything's messed up. We look at asthma. The data's there all over the place. Look at all the other diseases, peripheral vascular disease, obesity, inflammatory bowel disease, biliary disease, colon cancer. Microbiome has something to do with it. Economist Magazine, 2003, Shapes of Things to Come. We're evolving. We're evolving very well. Anyone seen these slides before? CDC data. These have been around for a while, but this is looking at obesity in the United States. The map of the United States is basically anyone with a BMI over 30, you're going to show up in a color. Right now we have 3 colors. White, which means no data. The light blue is less than 10% of the people in that state are obese. And then the next one up is 10 to 14%. I'm going to jump ahead really quick. 2000, bang. 14 years. Look what happened. Now you've got a whole new category. Half the country has. 7 years. Bang. 1 in 3. We don't evolve this fast. I'm sorry. Something changed. 2009, 2011, Colorado, they finally caught up to us. This is scary stuff. Anyone ever heard of epigenetics? I know some people have been here before to Randy Jirtle's lectures. When I first came and saw Randy Jirtle years ago, he's at Duke University, he's a radiation oncologist, and I heard him do a lecture on epigenetics. I was blown away, and it changed my life that night on what I need to learn. So I started learning this stuff. The study of heritable changes in appearance of gene or gene expression caused by mechanisms other than changes to the DNA structure. In other words, can your genes be read differently based on exposure to the external world environment? We're in the center for the environment. Stress, chemicals, food, drugs, blah, 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 blah. Human Genome Project. When they first embarked on the Human Genome Project, they said, we will have an unbelievable amount of genes. They were thinking 100, 150,000. Well, we had 24,000 genes. So like, okay, a worm must have maybe 500 then. A worm has 21,000 genes. How does a worm, that little creature that slithers along with almost no structure compared to us, have 21,000 genes and we have 24,000 genes? It doesn't make any sense. The analogy that Randy Jourl uses, which I think is fantastic, is think of the hardware of a computer and the software of a computer. The hardware does nothing until you put software in. Hardware is your DNA. That's your genetic code. That's your book of life in every nucleus, in every cell in your body. The software is the environment. That's the epigenome. That's every signal that you receive from anything in your life can alter your DNA. And we're going to talk a bit about this. Anyone ever heard of the agouti mouse? This is Randy Jourl's work. So the agouti mouse is a mouse that's bred to be yellow, fat, and diabetic. If you put mom and dad, agouti mouse together, they're always going to produce a baby's yellow, fat, and diabetic. Terrible thing, right? Well, during some research work, they looked at it and said, well, what happens if we give these mice methylators? And we're going to explain to you a little about this, and I'm going to apologize for science, but I think you all need to see this. Methylators are foods. There's vitamin B9, B12, choline, folic acid. Everyone's heard of folic acid. What does folic acid prevent in pregnant women for their offspring? Neural tube defects. Somebody figured that out. Why? Because it's affecting the methylation of the genome. It's affecting how we read. I do this all the time now. Since I've gone back into my fellowship, I spend more time on biochemistry. When I was in medical school, I will submit to you, I hated biochemistry. I thought it was the worst thing ever. But now that it makes sense from biochemistry where the rubber meets the road, and I could take that and then translate it into a kid getting better, I love biochemistry now. It's not this ethereal place. It's real. Right up here. B12, folic acid, you get the cycle spinning up there, and then up at the top you have SAMe. That's effectively helping your DNA get read appropriately, helps methylate the genome. What does that mean? Well, here's your DNA. Here's your chromosome. You're unwinding it. It's got this little protein called a histone. You keep unwinding it, and everyone remembers Watson and Crick, right? Here's your base pairs going all the way around. Me. Carbon atom, 3 hydrogens around it. Methyl group. Food gives you those. It puts a sticky note on there that says, don't read me. If you take that sticky note off, guess what happens? It reads it. So what happened? Well, that sticky note comes on here, and then all of a sudden you see this protein start to get built, and then you end up with this, and then the protein goes and does something. You saw in the video in the beginning of the show by Jeffrey Bland how the thing was crawling along, and the protein structures were conforming, and somebody's wrapping around it and sending the signal to the cell to do something. Proteins do everything in our body, function-wise. So it makes a difference which proteins we make. We make the wrong proteins, oops, we got a problem. So what happened to that mouse? Well, you see there's a brown one over there. That doesn't make sense. Why do you have a brown one next to a fat, yellow, diabetic agouti mouse? Well, because when they gave the methylators to mom during pregnancy, guess what happened? Her baby came out brown, skinny, non-diabetic. Holy mackerel. The first time we've seen that we can alter the DNA of the offspring. Now, we didn't change the code. We just changed how it was read so the kid came out normal. Think about that. That is profound. Food during a critical time during pregnancy changed the offspring. So it goes back to that slide about what's going wrong with our incubators. If mom is eating crap food at McDonald's, could be doing something. How do we study that? It's almost impossible. Nutritional studies in humans is a joke. It's very difficult because there are so many variables. And they always say, oh, you look at that study, it didn't prove anything. We got another study that proves this wrong. Epidemiology says if you eat healthy food, you're going to be good. We don't know what your health should be exactly yet. We're going to find out because as DNA analyses go on, we start looking at your code, comparing it to what you eat and what disease you end up with. We're going to know. I'm praying that someday soon we'll take a little prick of your baby's blood at birth and go, this is what he needs to eat. Kid lives forever. Joke, but seriously, I mean, something to that attune. I mean, it's almost Star Trekian. Dosage and timing is key. Critical times are in utero, birth to 1, puberty. You mess with those 3 times, you're getting the bull by the horns. Analogy with 1 or 2 glasses of wine is pretty good, but you drink the whole gallon, you're falling over and you're doing something you shouldn't be doing. Now let's go back. So Randy Jourl was in there in his lab, Dana Dolino. He says, well, let's see what happens. We'll give that mouse while it's pregnant. We'll give that mom those methylators, those good foods. Now let's give that mom bisphenol A. Everyone here's heard of BPA. That's that thing that was pulled out all those plastic baby bottles that were feeding babies forever. Well, BPA acts like estrogen. When you get BPA in your system, your body says, ooh, that's estrogen. Well, I'll tell you, I don't think I need any more estrogen. My wife might think I need a little more to be a little bit more feeling, but I don't need any more. But that BPA is out there. Credit card receipts. Every time you grab a credit card receipt, that's got BPA in it. Lining of cans, BPA in it, right? It's supposed to be banned. It's not banned yet. It's been banned in Europe and Canada for a while. We're taking it on sweet time. Well, so they gave them methylators and then they gave them BPA. What do you think happened to that baby mouse? It went back to yellow, fat, and diabetic. So now we have a push-pull in the environment. We have good food equals good. Chemicals can override the good food. Now we've got bad again. So now we have more multifactorial things going on. Now we have the gut microbiome as a player in obesity, because we know if you have more firmicutes, you're going to be fat. We know if you get exposed to chemicals, you can be fat. That's just two pieces. There's a lot more going on here. H. pylori cuts off your ghrelin. I mean, look at how many variables. Dynamic, amazing, crazy. How do you study all this? You don't. You go back to what did nature intend? What were we put on this earth to deal with? What was the original intent? Was the original intent to be delivered in OR? Was the original intent to suck on a baby bottle and drink formula? Was the original intent to get tons of antibiotics for ear infections? No. So if you just think about what we're doing, that's the problem. Let's look in 2000 U.S. food consumption. 10.6 is vegetables, fruits, nuts, and beans. Look at the ones. Grains, fats, oils, sugar, sweets. Going back to that enterotypes part, we know that what we eat dictates what bugs we get. Valproic acid, known to hypomethylate the genome, known to cause systemic lupus erythematosus, an autoimmune disease. So this drug gets into your DNA and takes off that sticky note. Good for seizures. Hope you don't get lupus. BPA, drinking while you're pregnant. American food. This guy's crazy. Cigarette smoke while you're pregnant is associated with asthma. Generation XXXL. I mean, come to my clinic. This is real. We have kids who weigh 100 pounds at 6 years old. I mean, insanity. You know, look at this thing. Fruits and vegetables have gotten more expensive since 1985. Even the sweets, fats have gotten cheaper. Why? Food, farm bill. Anyone who's come to this environmental center has seen lectures related to this stuff. It is a crime that we're allowing our federal government to continue to subsidize corn, soy, and wheat like they do and then make vegetables and fruits so expensive. And you wonder why food deserts exist and people in inner cities can't afford anything healthy. It's crazy. This is something I think is beautiful. I had the pleasure of going down to the research center in Kannapolis and sitting with a gentleman named Moshe Schiff a couple years ago who's done some work up at the University of Toronto Sick Children's. And I bring this up only for an epigenome discussion, and that it ties back into the gut with probiotics. But let's take these they're rats. And this is a study looking at nature versus nurture. Everyone always asks that question, is it the nature or is it the nurture? Which part is more important? Is it the chicken, the egg? Right? Well, we've got a biological mother here. She's a low licker and groomer. When her babies are born, she doesn't pay as much attention to them. We've got the adoptive mother, who's a high licker and groomer. So let's cross-foster these babies. So genetically, we're going to take a baby born to a low LG mom. So that means the baby probably is going to be a low LG genetic code. We'll take babies born to the high LG mom. We have a high LG genetic code. And let's cross-foster them. So we're going to see what nurture does. All right, and so now we're going to look at environmental programming of gene activity. So what we're talking about now is glucocorticoid receptor function of the gene. So glucocorticoid is your receptor that evolves with cortisol, right? So fight or flight. So you wonder why some of us are more stressed out with the same exact experience. How these guys go off to war, 9 guys in the same unit, same exact bomb goes off, same experience, 2 come home with PTSD. Probably some do their genetic code got shifted during nurture. Probiotics. Probiotic treatment of these rat pups normalizes corticosteroid release and ameliorates colonic dysfunction induced by maternal separation. This is a different study looking at when the mice were taken away from their mom, so the nurturing didn't exist. Their gene got all messed up. Give them probiotics, whoop, you fixed it. This is not showing, this is supposed to be a wailing baby. It's the environment, stupid. I mean, it really is. We do not pay nearly as much attention as we need to what our external environment is and how it affects us and what we need to do about it. This is not rocket science. You know, Everything I've set up here is easy to understand and not difficult to implement in your life if you so make the choice. If you don't make the choice, absolutely, this is difficult. And there are plenty of people in the world who absolutely refuse to do what's right for themselves and then expect the rest of us to deal with it. I submit to you there's a new paradigm for this. Renz et al. In Nature's Immunology in 2011 put this together. He said, susceptibility genes, organ or tissue-specific, cell-type-specific stuff. You have your environmental microbiota, you have your barrier microbiome, and you have food. You have your epigenetic layer, then you turn your immune system on. If you screw with these, you screw with this. If you mess with this, you will develop either disease or protection intolerance. This is Disease Management 2013. It is not pill for ill. It is, what did you do wrong here? How do we change this? How do we get back here? How do we fix the immune system? How do we stop disease and get back to protection? That is what health should be. Now, the problem is that our medical system doesn't allow this. 10-minute visits are very hard to teach someone to do the right thing. But lectures like this and going to read books about this stuff can help each one of us individually make these decisions. I'm just going to briefly talk about a little bit of inflammation stuff, and then I think we're going to be out of time. There's really nice data looking at when you have the wrong microbes in your gut and you eat the wrong foods, you actually release chemicals in the gut called lipopolysaccharide, or LPS. Gram-negative rods, specifically, there's certain types of bugs that we have that are in our gut. And if you eat a high-fat meal, you end up with inflammation. They looked at a study in 100 grams in Australia. They looked at a study of adult men eating 100 grams of kangaroo meat versus 100 grams of Wagyu beef. And for you guys, Wagyu beef would be American, corn-fed, marbleized, beautiful steaks. You know, think of Ruth Chris sitting there with this beautiful steak, juicy, dripping everywhere. That steak is an inflammatory nightmare. When they looked at the blood markers of these humans after eating 100 grams, which is a little more than 3 ounces, the kangaroo did nothing. The blood was perfectly even. Because what? That was what nature intended. Nature intended you eat an animal running around. It said, take your bow and arrow, shoot that thing down, and then cook it for the whole village. It didn't say, grow it, and take that cow that's supposed to eat grass with 4 stomachs to ruminate it all, and then feed it corn to make it fatter so we all love it more, and then eat it. So the blood markers of cytokines, which are these chemicals that produce inflammation, went through the roof. So you wonder why guys go into a restaurant like this and have a heart attack after their meal. Because you just flooded your system with inflammatory chemicals that said, clack, shut. They also looked at other work, and they said red wine. Why is red wine good? Well, it's got resveratrol in it. We've heard of that. It's a chemical that helps us prevent telomeres, which are these things at the end of our DNA, from disappearing so we don't age as fast. But it also has an effect on inflammation. Olive oil, anti-inflammatory. Tons of minerals, tons of herbs are anti-inflammatory. You've heard of curcumin. You've heard of holy basil, ginger. These are all these foods that people cook with. These are spices. Why did people use spices in cooking? Does anyone know? Taste is a secondary reason. The original reason was anti-infectious. If you put spices in foods, they don't degrade as fast. Bacteria can't grow as much and you won't get sick. So that was the original reason for spice. Spice was the #1 expense commodity back in the early ages. Gold was nothing compared to spices. Spice trade was it. Because if you had spice, you had food that could last for a while and you could eat. Spice actually is good for us. And American food is what? It's devoid of spice. It tastes, yeah. We got fat in it. Yeah. We got salt in it. We don't have much spice. This was one of my ramblings one day. I have a whiteboard in my office and I get crazy sometimes. And this was in 2009 when I sat here and was trying to figure all this stuff out in my head. So I wrote it all down and I came up with this paradigm as I was going through some of this stuff. And I said, this is disease, morbidity, and death. What is the influences of that? Common themes of disease: poor nutrition, decreased vegetables, fruit, fiber, decreased whole food, increased flour, sugar, seed oils, animal protein, and dairy. Decreased sun exposure, vitamin D, Treg cells, dampening your immune system. Decreased exercise. Your bone talks to your pancreas. There was another lecture I did a long time ago that was fascinating. When you run, you actually send signals to your pancreas to do a better job of dealing with insulin. Whoa. Decreased psychological support with increased stress. Overclean environment, chemical exposure. All these unbalanced things send this whole cascade into disarray. The only thing in this entire slide that is static is the DNA. That's the only thing that's static. It never changes. Only how you read it changes. And that's all because of the environment. There's a book called The Blue Zones by by Dan Buettner. He's a National Geographic photographer, and I sort of stole his book and wrote my name over it, but really, truly, it's his work. And what Dan Buettner said is, where in the world are the most 100-year-olds, and why are they there? Centenarians. And so he looked around the world and found that there are places, Okinawa in Japan, the Seventh-day Adventists in La Jolla, California, the island of Crete. In Ecuador, there's a village that has a lot of 100-year-olds. And what was the common theme? There had to be a theme. We don't need double-blind, placebo-controlled studies to figure this out. What were these people doing? They ate plants, and a lot of them. They avoided chemicals. They had natural exposure to bugs. The microbiome got fed. They had a sense of community, purpose in life, nurture. It affects your genome. You're around good people, like this room, high energy level, everyone in here is positive and good. This is a good room. It feels good to be here. You get in a room with a bunch of prisoners, that ain't a good feeling. It'll affect your DNA. Judicious sun exposure, daily exercise, eat 20% less, maintain a healthy weight. Skinny people live longer. The only downside is if there's a famine, I'm gone first. Spiritual practice, slow down, work less, rest more. That's the anti-American philosophy there. It's really unknown today if and how your child is affected by your particular environment. If they have a genetic makeup that doesn't handle a particular stress well, then they will likely suffer disease. The future will allow us to know in advance through gene analysis who is at risk. Well, right now, and obviously you can see this was a slide from last year, it is 2013. It is about balance. And that balance is the key. And if we can figure out the balance for our genome and our DNA, we figured the game out. Knowing that we are unable to know if our child is at greatest risk, we need to act as if they are. The precautionary principle of Europe states that if we don't know if it's safe for us, we shouldn't release it into the environment. The United States doesn't have that. We have 86,000 chemicals released without any data knowing whether they're good or not until we find out that they've messed up a whole generation. We need to be cautious beforehand and not wait till it's too late. And that's the end.